Okay, so we'll get started. So I'll just do a little introduction. So welcome everyone to the My STEM Future. Just to give you a bit more about how the session is going to work, if you haven't used Zoom webinar before, we've got a chat box, which you should see hopefully at the bottom of your screen or at the top, that you will be able to ask questions for our lovely scientist who's going to talk to you, for you guys. You can raise your hand as well for you to talk that way. There's also going to be some live polls that you can answer as well to get your point of view. And if you want to catch up with any of our previous talks, they're on our website, mystemfuture.org. And you can find us on all social media platforms as well. That's all from me. So I'll hand over to Dimitrios. So welcome, Dimitrios.
Hello, everybody. Let me share my screen. Great. My name is Dimitrios. I'm a postdoc in the Department of Medicine at the University of Cambridge, and I'm very happy to be here. Welcome to all of you, and thanks a lot for the invitation. It's a great initiative. I would like to talk to you a bit about my work in cancer research, which focuses on the use of genome editing technologies to identify ways to harness our immune system, to use all the tools that the immune system has to fight cancer, which is called immunotherapies. At the same time, I would like to give you kind of a general overview of how I ended up working on that subject, and hopefully that could be helpful to your future decisions as well. So why did I choose this field in the first place? Growing up, I had a number of scientific heroes. Some of them are real, some of them less so. And I'll show them to you. And if you can write on the chat box whether you can recognize any of them.
I might not know the top right one. Ooh, that's bad of me.
Should we give it away?
Yeah, who's the top right?
So top right, Marie Curie, the 2 Nobel Prizes for important discoveries in radioactivity. And also from the real ones, you may recognize Charles Darwin for...
Yeah, we have that in the chat box.
Yeah, great.
And Dr. X.
Yeah, Professor X. Then Rosalind Franklin for co-discovery of DNA. Iron Man, you may not see it very well. Bruce Banner, the Hulk. And also Jane Goodall, a famous primatologist, with really important work on the on social structures of the great apes. So moving on, when I had to choose, I guess, what to study, I chose biology since I was always quite fascinated by it. I come from Thessaloniki, a city in northern Greece, so I actually studied there for my bachelor's degree at the Aristotle University of Thessaloniki. And the cool thing about my degree was the scope was quite broad. So we did molecular biology and genetics, but at the same time, zoology, ecology, botany. So we're exposed to pretty much a full spectrum of what biological sciences have to offer. And even though I didn't choose to go towards zoology, ecology and stuff, it was still like really interesting to learn about those things. Also, most programs, I think nowadays, undergraduate programs, including my own, gave us a chance to spend some time in a lab. So that was the first experience working in a lab. But I actually faced quite a lot of difficulty adapting, going from high school to the university, adapting to a more academic type of studying. The material needs to be approached in a different way, but I was very fortunate that essentially the solution to that was kind of spending some time in the library with some friends, and we're exchanging tips and kind of inspiring and giving ideas to each other of how to tackle the coursework. So that was really helpful. After that, and during my undergraduate degree, I had the chance to be a member of the Erasmus program and spend a semester in CRC in Paris. That is something I would like to stress out, that research actually gives a lot of funded opportunities. Maybe not the Erasmus program anymore for the UK, but there are a lot of funded opportunities that would allow somebody to travel and spend some time in a different university, which is, I think, is extremely meaningful. During my time in Paris, I had pretty much half a year of an extended research project. And that was really useful. That was quite difficult in the beginning because I had to adapt to working more independently and also in a foreign country, in a foreign language. But then again, it was mainly through actually making some good friends in the lab and having good mentors that you know, helped me take my time and find my own way of dealing with those issues. Finally, when I finished my undergraduate degree, I wanted to first to travel and second to get some more technical and hands-on laboratory experience. When you do an undergraduate degree, for the most part, even most programs have a big part of it that is kind of practical, like on the to be able to spend time in a lab. You focus a lot on theory, and it's things that you need to learn to start understanding how things work. However, before I proceeded to further studies, I wanted to kind of focus a bit more on the technical skills. So I managed to get a job as a research assistant. I moved to the USA, to New York City, to the Rockefeller University, which was a very meaningful experience for me. The level of multitasking that I needed to develop was very different to what I was used to. Also, the hours were quite long and there was a lot of pressure as well. And it was kind of the first time that I had to see biology as also how you could feel doing that as a job rather than as a field that you study. After that, I moved to Cambridge, where I did my MPhil, my PhD, and now postdoc at the Sanger Institute and then the Department of Medicine. And my focus was, I knew that I wanted to work on cancer, but from my experience in the US, I really liked immunology as well. So like the field of cancer immunology seemed like a perfect match of the two. What I would have to say about the PhD and the MPhil is that it can have a lot of ups and downs. Like when you wait for an experiment to work and it doesn't, it can be quite difficult. At the same time, there is like complete elation when sometimes you get the result you were hoping for and you see it after many tries. But overall, I have to say that I made some great friends in the process and had a lot of fun, and it was definitely some of the happiest and the most fun times of my life. So let's move to what cancer is exactly. So according to Harland Varmus, he described it as a distorted version of our normal selves. Varmus is a Nobel laureate and cancer researcher. And if you would like to read a bit more about the history of cancer as a human disease throughout human history, there is this great book, The Emperor of All Maladies, written by a cancer researcher and author Siddhartha Mukherjee, who is at Columbia University. And it's truly a great book. So if you're interested in the subject, just, yeah, you can easily find it. So cancer could be described as a disease of chaotic growth and disobedience. You can think of the cancer cell as a rogue cell, as a maverick cell. Usually the body has a very tightly controlled system of principles within the cell that tells the cells how to operate and when. And this sort of balance at the level of the cell and the organism we call homeostasis. Essentially, cancer is a disease that keeps of a cell that keeps growing and doesn't pay attention to those rules. Also, the cancer cell has the ability to deceive and evade our immune system, which has been evolved to protect us. And also it can evade all a lot of the therapies that we throw against it. But since cancer starts from a normal cell, it's very similar to it. So it poses the difficulty in distinguishing self from non-self, which is a big issue when it comes to cancer therapy. Actually, at this point, we can do a poll that based on those things that we discussed and based on what you know in general, can you choose phrases that describe the cancer cells?
So we'll give you a few minutes to do that. And it's also multiple choice, so you can choose a few. See what people come out with. Okay, so the answers are, so move to distant tissues, metastasis, lose their self-destruct mechanism and grow indefinitely. Do they ignore homeostatic regulation and do not respond to signals?
Yeah, great.
And
great answers.
are not specialized?
Yeah. So yeah, you nailed it. So what we can see, as you very accurately said, so the normal cells multiply according to the strict rules of homeostasis. However, the cancer cell ignores this regulation and does not respond to signals from other cells. The normal cell is localized where it's supposed to be to perform a very specific function. However, the cancer cell, as you can see actually here, as the normal cells have these molecules on their sides so that they can connect them to the other cells into the tissues where they're supposed to be, the cancer cell loses that. So it can actually go to a distant place in the body and start growing there, which is what we call metastasis. And this is one of the most difficult things to treat when it comes to cancer therapy. The normal cells differentiate into more specialized types because they have a very specific role. So they're specialized to perform that role. However, cancer cells lose this specialization. And their main target is survival and growth. Also, normal cells, a very important aspect is that they have this self-destruct mechanism, which is a mechanism that we term apoptosis, and essentially means that if a cell receives some damage to its DNA or because of aging or other factors, essentially the cell would die to protect the organism from cancer. However, the cancer cells, they lose the self-destruct mechanism, as you very correctly said, and grows indefinitely to form either solid tumors that you can see what it would look like in this sketch, or leukemias, which is essentially cancer cells floating in our bloodstream causing blood cancers. But then what are the causes of cancer? Can you write in the chat box? I'm wondering what based on what you've heard, what do you think that are some of the common causes of cancer?
So if you're in the chat box, just add what you think are some of the common causes of cancer. And then we've got a couple of questions. So we've got, what is homeostasis in more detail? That word seems quite strange.
Yeah, it is. That's a very good question. So homeostasis is not one single thing, but you can think about it as a general balance within the body. So a cell on its own, it's not an organism. An organism is an accumulation of these cells that work together, and different cells that have different functions and perform different things. But we need all of them for an organism. Think about the human body, for example. For the human body to function properly, you need muscle cells, you need neural cells that would respond to specific stimuli that can send signals to other cells. You need your cells of your immune system, the cells of your heart. And in order for both the organism and the cell itself to be able to function, it needs to follow those rules. So it has this inherent programming, that, if you will. So this is what homeostasis is this balance which is dictated by a number of rules that are written in our DNA that would allow the body to function and also protect us if a cell wants to go rogue and become cancer. It is a fancy word to think of like general balance in the cell and the organism.
That sums it up in one word.
Mm.
Yeah, so we've got one answer for what are the causes of cancer? So we've got chemical exposure.
Very nice. Yeah. Should we move on?
Yeah.
Yeah. So yeah, that's actually, as you see here, some chemicals and pollutants can cause cancer. That's true. Also, some viruses can cause cancer. UV radiation can cause cancer. Smoking, as you probably know, eating a lot of processed foods or excessive alcohol consumption, maintaining an unhealthy weight, obesity is related to cancer. However, yeah, that is something to discuss, because if in another time, maybe, because obesity is also genetically determined to a great extent, but not only. Also hereditary traits, which is essentially traits that we get from our ancestors, and also random mistakes in DNA replication. So we're going to do another poll. If you can think of which of these can be preventable and which not.
So there we go. Hopefully you can see the poll now. It's also a multiple choice. So which of these do you think is preventable? Now we've got a question. Why does COVID-19 treatments seem so close to cancer treatments?
That's a good question. So there are a lot of some of the treatments for COVID are antibodies, which are produced by some immune cells in our body called B cells, and they target specific molecules in the kind of, if you will, in the enemy cells. So antibodies, B cells can produce antibodies either against an infectious disease, against the virus, for example, but we also use it in cancer treatment. So we use antibodies, and we will actually talk about that. This is a very good question a bit later. So antibodies is something but it target different things. In the case of COVID-19, they target the virus so they can neutralize it. In the case of cancer, they would have a bit of a finer role to target either the cancer cell or some immune cells, and kind of reprogram them to allow the immune system to wipe out the cancer. Also, for cancer, we use chemotherapy, which is not something that would be very advisable to use for COVID-19 treatment, because that is a bit different. But it is true, there are some immune molecules that are used for both cancer and COVID-19.
That's good. So we knew a bit more about COVID-19 then, even though it was a surprise.
Yeah.
So we've got the answers. So we think that the following are preventable.
Yeah.
The radiation.
Good.
Processed foods.
Yep.
Excessive alcohol consumption.
Mm-hmm.
Unhealthy weight. And smoking.
Right. Yeah, that's very, very good. With some two kind of question marks that I would add. I mean, UV radiation, you can put sunscreen, for example, and you can avoid the sun. I don't think necessarily would be very good for our health to avoid the sun completely. So some amount of UV radiation you cannot avoid, but I would consider to the great extent a preventable cause of cancer, because you need quite to be like this constant exposure, to be not careful at all about the sun, which most people thankfully nowadays are. And also the unhealthy weight aspect, that even though of course you can exercise and trying to maintain a healthy weight, but this is also it's a trait that is it's prescribed in our genes to some extent. So for some people, even though for many people it could be a choice, for some people it is not a choice. So for some people it could be way harder to maintain a healthy weight than for others. So just something to keep in mind. But you can hope that you can avoid chemicals and pollutants, and also you can get vaccinated for the viruses, for a few of the viruses that could cause cancer. However, as you very rightly identified, that random mistakes in DNA replication and what you get from your ancestors is something you cannot really avoid. And what this would lead us to think is that cancer is, no matter how horrible it sounds, it is a naturally occurring phenomenon. Thankfully, though, our immune system has evolved ways to protect us from it, because cancers happen in our body. The cells trying to go rogue happen all the time, but our immune system is there to wipe those cells clean. So let's go and talk a bit about the immune responses against a tumor. So a tumor would start from a cell that has received, that is going out of control, is going rogue, as we said, and it multiplies, forming a tumor. Can you write on the chat box whether you've heard of any immune cells that you may have heard either in previous talks or from other sources that you may think that they could play a role in this crosstalk between cancer and the immune system?
So this should be a really good test for who was paying attention last week, because Amar did mention some different immune cells. If you think of the bullets that he had on one of his slides.
But don't worry, I won't tell Omar.
If you don't get the right ones. We've got a question as well, though. So how to read our DNA and find out homeostasis rules in a body, while the higher risk for a specific person?
Yeah,
So,
that's an excellent question. And I think the answer to that is pretty much everything we do in biological science. Essentially, if you go to the core of the question that's most biologists are trying to answer, regardless of the field of the disease or of the mechanism, we're trying to figure out what is this set of rules within our body, and we can read the DNA. As you may know, the DNA is essentially, you can think about it as the book of life, like everything that describes how we are and how we're built, essentially, how we've ended up being through evolution, is in our genes. And we can read that with sequencing technologies. DNA sequencing, for example, is a way of reading that book. However, in order to make the link between what the book says and what is the outcome that we see in the body, that is actually quite hard. So I wouldn't have a very specific answer, because it's essentially all the experiments that different researchers do try to address to some extent this question: what is a normal homeostatic kind of control, regulation? What are the principles in the context of health and in the context of disease as well? So yeah, I don't have a very specific answer to that, but it's essentially that's a very core question that many biologists are trying to address through different technologies. But reading the DNA would be through sequencing technologies.
that's brilliant. They say, thank you for answering the question, but we don't have anyone naming any immune cells. So I think we might have to tell them. Oh.
It's okay. We can go ahead. They do have very weird names.
They do.
So let's say that the tumor happens in the body, and one of the cell types that can go against the tumors is the ones that are called natural killer cells, or NK cells. As the name implies, they're very good at killing, and what they kill is either tumors or cells that have been infected with a virus. So it's all about protecting us, right? To wipe out the cells before they spread or before the viruses start infecting more cells. And they do that by secreting, by releasing to the tumor cells this cytotoxic substances that we call perforin and granzyme. Another type of cell is the CD8 cytotoxic T cell. I believe Omar may have talked about that as well. There are similarities in the way they operate and the way they kill cancer through secretion of cytotoxic substances, but also they have differences in the way they're programmed. Some other cells that are called CD4 T helper cells, they work not necessarily by attacking the cancer itself, but kind of by enhancing the immune responses of other T cells, such as the CD8 cytotoxic T cells. And also another category of immune cells, the macrophages, they also secrete these substances that are termed pro-inflammatory cytokines, which kind of boost the function of other cells around them, such as T cells and NK cells, while at the same time, they can also attack the tumor as well. These are quite complex processes, and there is overlap, but this could be like a very kind of basic way of summarizing this. But let's focus a bit more on the interaction between a cancer cell and a cell of the immune system. Let's take the case of a tumor cell and a CD8 cytotoxic T cell, for example. What science has discovered is that the tumor cells, being messed up as they are, they express some abnormal proteins, which is great because these can be recognized by our immune system as foreign. If you remember from the beginning of the presentation, I mentioned that one of the main difficulties is telling the difference between self and non-self when it comes to differentiate between what is a tumor cell and what is a normal cell. So that happens, and the cells have this mechanism that these proteins are presented through a process that is called antigen presentation to the immune cells. And this is great because that is part of the process of how the immune cell would identify what should be its target and which cell to kill. However, the tumor cells express these molecules that are termed immune checkpoints. And what they do is that the thing that you see, the red one here that binds to this receptor that exists on the immune cell. And what it does is that actually it's a signal that the cancer cell gives to the immune cell and tells it to just get deactivated. So through this process, the cancer cell manages to become invisible to the immune cell. Think of the immune checkpoints like the cancer cell's invisibility cloak. That results in the immune cell getting confused and not responding properly. However, scientists have figured out this mechanism, and what we can do is that we can design antibodies, which are these Y-shaped structures here, which are blocking the immune checkpoints on the tumor cells and their receptors on the immune cells. And by doing that, essentially, we're taking away the cancer's invisibility cloak. The immune cell wakes up and realizes what is going on, and then it responds quite aggressively by secreting the cytotoxic substances that we talked about earlier. The outcome of that is that the tumor cells die and we have tumor cell death, which is obviously great for the patient. And this approach is kind of one of the basic principles of immunotherapy. How can you tweak the immune system, how you can take away cancer's invisibility cloak and allow all this incredibly complex machinery that our immune system has to attack and wipe out the cancer itself as it has been evolved to do. So immunotherapy is a revolutionary cancer treatment, and to convince you about that, I've picked the example of metastatic melanoma, which is a type of skin cancer. Metastatic melanoma was a cancer that the prognosis, for which a prognosis was very poor, and the disease was unfortunately almost always fatal. Before 2011, the standard treatment for that was chemotherapy, which had on one hand a lot of side effects, but on the other hand, quite minimal advantage to the patients. However, when immune checkpoint blockade therapy arrived, especially with a combination of two immune checkpoint blockade therapies, that disease is transformed, and now it has a 4-year survival of 46 to 53%. So we go from 100% lethal to almost half of the patients getting cleared of their tumors, which is absolutely fantastic. And the reason that you see this 4-year survival is that many times in cancer research, we look at for statistic reasons, kind of, okay, what is the number of patients that get diagnosed with the cancer, and then after 4 years, they're still alive, or after 4 years, the cancer hasn't come back. You may ask what happens if the cancer comes back from year 5. It is true, but it's a metric that we use in cancer research in the clinical world, and that is actually a very substantial result. However, not all patients respond to immunotherapy. Overall, different cancers respond better than others, but overall the response rate is around 20 to 50%. So at best, you still have half the patients not seeing any benefit from it. And also the vast majority of immunotherapies, they're targeting very few checkpoints. So we're quite in the beginning of this journey. However, the fact that it works for some is very encouraging. And what we need is to identify more immune checkpoints, and especially immune checkpoints that can be immune checkpoint therapies that can be administered together, so that they have an kind of a combined effect that would be similar, even better, to the one we just talked about skin cancer in the previous slide. So how can we discover more of these therapies? Well, as we said, everything is written in our genes. So that's the case for cancer as well. So what we can do is to scan the cancer genome, so the DNA of the cancer, for therapy targets. And to do that, I would have a question for you as well. So how do we figure out how a gene works? What do you think?
So just answers in the chat box again. So how do you think we figure out how a gene works? A machine is one of the answers.
Right. Yeah, sequencing machine could provide some of the answers. But what would you think that is a basic principle? If you have something in your DNA that you would like to see how it works, can you have any other guess? But it is true, we would use a lot of machines, and that wouldn't be possible without them.
Thankfully, I mean, before how they sequence the human genome, that was,
a lot of machines.
Data strip.
Yeah.
yeah, a lot of. So we've got another answer, a data strip.
Okay. No, these are good answers, because it definitely, these things could be incorporated into the. But what I meant mostly, this is quite an open question, I guess, but the basic principle in biology in general, when we want to see how a gene works, the concept is like break it and see what happens. So disrupt its function and see what happens to the cell and to the organism when the gene is not working as it normally would. And for that, we're very fortunate to live in the era, science-wise, that we live, because we have the CRISPR-Cas9 system and a genome editing technology. I don't know, have you guys heard of CRISPR-Cas9? Can you raise the hand if you have?
Hopefully so, if they remember the talk last week. A few. So someone's heard of it a little bit. Okay,
great. Well, let's do a review of that. So the CRISPR-Cas9 system is composed of two parts, the orange and the yellow that you see here. The orange part is Cas9, which is an enzyme. You can think of it as a type of molecular scissors. And the yellow part is the guide RNA, which essentially targets the Cas9 protein to a specific gene. And it does so because it has similar sequence to the DNA that it wants to target. So based on that, the Cas9 and the guide RNA are recruited to a specific gene. And what they do is they cut. By forming this cut, you have what we call a double strand cut on the DNA, which means that the two strands of the DNA get separated. And what the outcome of that is that a lot of repair mechanisms within the cell get activated, effectively to try to paste back what was cut. However, this is done full of mistakes. And that results in the disruption and deactivation of the gene. So for the purposes of this, of what I would like to think of you, I would like you to think of Cas9 as this system that is targeted to a specific gene. It cuts the gene and results in its deactivation. This is something, when a gene is deactivated, we call it like a knockout. So when I'm talking about knockouts, I mean a deactivated gene, a gene whose function has been disturbed. So how do we can use that to our advantage to, as we said before, to scan the genome? So we do that with a method that is called CRISPR-Cas9 screen. And a screen works as follows. We use viruses to pack all the different components of CRISPR-Cas9. So we have the guide RNAs, and we also deliver Cas9 to the cells. We use those viruses to deliver the guide RNAs and the Cas9 to the cells. And that has as an effect that each guide RNA would target a different gene and knock it out. So we have this disruption in a gene. And what you see here from the different colors is that, let's say that the green cells have a knockout in gene number 1, the purple cells in gene number 2, the red in gene number 3, and so on. When you let this, and we do that on, let's say, on a dish in the lab, in a pool, in a culture of cells. What happens when you do that is that some gene knockouts give an advantage to the cells. So if you look carefully, the red ones, for example, you have only 3 here, but they grow quite a lot more after the passing of time. However, the yellow ones disappear. And why would that be? Is because the gene that it was targeted was essential for the survival of the cell. So the cells that have this knockout died. And some others, you don't see that they change much. For example, if you look at the green, they're still green before and after. And we do that, as you've very well said, is with machines, with this really fancy next generation sequencing machines that can count essentially what's the amounts of cells, and you can quantify whether a specific knockout made a cell multiply or die or didn't do much. So this is the basic principle of the screen. And to focus a bit more on the screens that I've done for my studies is that, let's say that you have this pool of cells in which some gene knockouts made some of the cells multiply more than others. So it gave them an advantage. For some, it made them a disadvantage since the cells with this knockout died and are not here anymore. And some gene knockouts didn't do anything. So what I was doing was I was adding immune cells to that equation. And through this next generation sequencing technologies, we could identify whether a specific genetic knockout, whether a specific knockout in a cell kind of confuses the immune cells and works as an invisibility cloak for the tumor cell, or whether another knockout essentially has the role of removal of the invisibility cloak and results in the immune cell getting activated against it. So making this connection about which knockouts do what is kind of the main aim of my previous work. And through that, we can identify that some knockouts make cancer cells more visible to the attacks of the immune cells. And essentially taking away cancer's invisibility cloak. And that is what can very well be used in the context of an immunotherapy. So just to approaching the end, I would like to give you like the full quote from Harland Varmus. This is from when he received his Nobel Prize, and he said, I believe he describes really well the process of cancer research in a very poetic way, and where we are and where we're going. So he said, we have not slain our enemy, the cancer cell, or figuratively torn the limbs from his body. In our adventures, we have only seen our monster more clearly and described his scales and fangs in new ways that reveal a cancer cell to be, like Grendel, a distorted version of our normal selves. And he references here the monster Grendel, who appears in the epic poem Beowulf, who is the legendary hero. Beowulf finds the monster Grendel. And Grendel was described like a distortion of a human being. And so Varmus makes a very poetic connection between Grendel and the cancer cell being kind of a human-like monster itself. But what I actually want to close with is a story that illustrates really well that the future of cancer research and cancer immunotherapy is a very promising one. So I would like to tell you the story of Judy Perkins, who is a 49-year-old woman that was diagnosed with metastatic breast cancer, which had spread around her body. She had received multiple treatments, multiple chemotherapy and radiotherapy, which had not managed to stop the cancer. She was given only 3 years to live. And she said the following, that my condition deteriorated a lot towards the end, and I had a tumor pressing on a nerve, which meant I spent my time trying not to move at all to avoid pain shooting down my arm. I had given up fighting. Judy Perkins received a personalized combination immunotherapy as part of a clinical trial, and the outcome was a staggering, durable regression. Ever since her recovery, she has backpacked the Appalachian Trail and has circumnavigated Florida by sea kayak. And she has resumed her very adventurous life and lived with her family. In 2001, she reached the 5-year cancer-free milestone, and you can look her up on social media. She's still giving talks and is in great shape after all these crazy adventures. So what was given to her was this really advanced immunotherapy through which they used next generation sequencing machines to sequence the DNA of the cancer and identify specific weaknesses like the Achilles heel of the cancer. At the same time, they sequenced her own immune cells and found that there were some immune cells that were good at recognizing the cancer, but for other reasons, they were silenced through the mechanisms that we described before. So what the researchers did was to extract those immune cells and multiply them in the lab to great numbers and give them back to her alongside immune checkpoint blockade therapy. And that is one of the most staggering results, because she essentially recovered from aggressive metastatic cancer. And in her words, I had resigned my job and was planning on dying. I had a bucket list of things I needed to do before the end, like going to the Grand Canyon. Now I have gone back to normal everyday life. And I think that's, to me, that summarizes the reasons why we do what we do, and why I find this field of study so incredibly inspiring. Thank you very much, and I'll be happy to take any questions.
Fantastic. Thank you, Dimitrios. My mind's blown. I don't think I've ever... I just think it's amazing, the immune cancer cells being able to use an invisibility cloak to hide from the immune system. It's just amazing.
Yeah,
Incredible. So we've got a question about why do you call it a knockout? Why is that name used?
Oh, that was pretty much like all terminology that stayed with us. Essentially, you knockout means deactivation. It means that you disrupt the normal function. The gene doesn't work anymore, and it can be done with different ways. But with CRISPR, you can do that at what we call a genetic level. So it kind of essentially edits the DNA of the cell to mess with the function of that gene. And essentially, think about it like in boxing, like a KO. It's out of action.
That's what they actually refer. They think that it's in boxing. So when they're hearing about a knockout, so you're very right, Jorge, about thinking about that.
Yeah.
And then the same attendee says about the Johnson & Johnson called Blue Night Challenge. Have you heard of that one?
I'm afraid I haven't. What is it?
It's a Blue Night. This is a topic host-directed therapeutics that restore homeostasis to the host, even if they've infected, to improve directionality towards homeostasis. So that's quite interesting. Okay,
I actually haven't heard of it, but I'll be happy to look it up, and if you want to shoot me an email, I can look it up, and I can write you an email to all of you if you want, to continue the discussion. But yeah, I'm afraid it sounds really interesting. I will definitely look it up.
Sounds interesting. Wonder how the body can return to homeostasis if it's got an infection. It's like, okay, no, we're actually okay. No, you're not. And then again, just so does sepsis have to do with the response? So with something you said earlier, is it similar to sepsis, would you say?
Sepsis, no, I didn't talk about that. That could be caused by different infections. But for example, all these diseases that you mentioned, you can very well describe them as a disturbance of the homeostasis, because you have essentially the regulatory functions going out of control. So sometimes, you know, a cell needs to die to protect the rest of the host. For example, a cell normally should be able should go through this path of self-destruction if it's infected with a virus, or so that it prevents and essentially trap the virus in that, sacrifice itself and trap the virus there so that it doesn't spread. That will be a homeostatic control, that the body is like, okay, how can we maintain our overall balance? We need to sacrifice some cells. So the cells are like, okay, I'm gonna do it, self-destruct, take the virus or the cancer with me.
And those cells just regenerate as well? Or
Well,
sacrifice themselves?
It depends on the cells. There are cells that regenerate. We have stem cells in our body that are able to, which are kind of the storage, the safety that we have, and they can differentiate and produce more of the different cells of the organism. In our adult life, some cells, like the neural cells, for example, they do not replicate as much. So that's why it's quite hard to treat. Or like muscle cells, you know, if somebody unfortunately has an amputation, for example, that you don't regrow a limb as lizards would do, for example.
So no evil scientists becoming like the Dr. Lizard with regeneration. Damn.
Exactly. Yeah. No.
So
Unfortunately
hopeful.
not.
Brilliant. Well, thank you. I think that's all the questions that we have. So we'll let everyone get back to their evening, especially you. So thank you so much for that talk, Dimitrios. I loved it so much.
Thank you. Thanks for inviting me. I had a great time. Thank you very much.
Yeah. Well, have a good night, everyone. We'll restart our MySTEM speaker series in September to hear us again, but enjoy your summer breaks and holidays and all the sunshine.
Use sunscreen though.
Yeah. Goodbye, everyone.
Bye-bye.